curious minds to another deep dive. Have you ever stopped to think about those incredible hidden blueprints tucked away inside every single cell in your body?
Oh, chromosomes, yeah. Amazing structures.
Exactly. They hold all the instructions for, well, everything that makes you you. And getting a handle on how scientists actually read these tiny, complex things, it unlocks some profound secrets about our health.
It really does. So today, we're diving deep into cytogenetics. That's the science of how we actually, observe these vital blueprints, how we spot changes.
And crucially, what those changes mean for human health and development, right?
Precisely.
Our mission for you today, as we explore this field, is to walk away not just knowing stuff, but really getting the why behind these powerful diagnostic tools. Imagine getting like a high-res snapshot of your entire genetic instruction manual.
That's pretty much what we're talking about, yeah. We'll cover everything from getting that basic chromosome group photo.
Karyotyping,
Exactly. All the way to advanced techniques that can pinpoint really tiny genetic missteps. And then, yeah, we'll get into what happens when those blueprints are off, the impact of those changes.
Okay, so let's kick things off right at the beginning. How do scientists even see these microscopic blueprints? What's the first step to getting that full picture?
Right, that's where karyotyping comes in.
Right.
Think of it, like I said, as the ultimate chromosome group photo.
A group photo. Okay.
Yeah, it's basically an organized image of all the chromosomes from a single cell. But getting that perfect shot involves a specific process.
Right. It's not just grabbing any old cell, is it?
Not at all. Scientists take a cell sample, sure, but they specifically wait. They wait for the cell to be at a particular phase of division called metaphase.
Metaphase. Okay. Why that specific phase? What's special about it?
Well, in metaphase, all the chromosomes do something really helpful. They condense, get thicker, and they line up perfectly right in the middle of the cell.
Wow.
It's kind of like athletes at the starting line, all neat and tidy, makes them super clear and much easier to photograph accurately.
That makes perfect sense. So you get the photo, then what? They arrange them.
Exactly. Meticulously arranged, usually by size, starting with the biggest, chromosome 1, all the way down, plus the sex chromosomes.
So what's the big payoff? Why is this karyotyping still so vital?
Oh, it's fundamental. First, it gives us that clear picture of what's normal for an individual, or even a whole species, the 23 pairs we expect in humans.
But more importantly, for diagnosis.
Yes, much more critically, it's the main tool for spotting what we call chromosomal disorders, large-scale ones.
Like any extra chromosomes or missing ones.
Precisely those big changes. That's why karyotyping is still absolutely essential in genetic screening and counseling, definitely in prenatal testing. It gives invaluable information.
It's kind of amazing to think about. Imagine, getting that kind of snapshot of your own genetic makeup. What kind of insights could that give you about your biology? Something to mull over. So, okay, that basic snapshot is great for the big picture. But what if you need to zoom in, see finer details on the chromosomes themselves?
Right. That's where banding techniques come into play. Karyotyping gives the overall view, but banding is like adding a high-definition filter. It highlights specific regions along each chromosome for a much more detailed look.
How does that work? How do you make these regions stand out?
The most common way is called G-banding. It uses a specific dye, Jimsa stain, and this dye creates a distinct pattern of light and dark stripes, or bands, on the chromosome.
Stripes, like a barcode.
Exactly like a barcode. That's a
Yeah.
perfect analogy. It stains the tightly packed, mostly inactive parts dark, and the looser, more genetically active parts lighter. Each chromosome pair has its own unique barcode pattern.
Clever. Are there other kinds of banding?
Yeah, there are variations. Q banding is similar, but uses a fluorescent dye and UV light, highlights different regions. Then there's R banding, R for reverse, which does pretty much the opposite of G banding, light inactive regions, dark active ones.
So just different ways to look at the details.
Exactly. Different perspectives, different layers of information, helps confirm things or spot subtle changes within those bands.
Okay, so dyes give us detail, but then things get even more sophisticated, right?
Oh, absolutely. Now we're getting into techniques like FISH.
Yeah, sure. Hash. Like the animal.
No, it's an acronym. Stands for Fluorescent In Situ Hybridization. It's a molecular technique, and it takes specificity to a whole new level. What's really fascinating is how it works.
Okay, tell us. These genetic searchlights, how do they operate?
So instead of a general dye that stains everything, FSH uses these specially designed DNA probes. And these probes are tagged with fluorescent molecules they glow.
Tiny glowing tags.
Okay. And each probe is engineered to be complementary to, and therefore stick to, a very specific DNA sequence, maybe a particular gene or a region known to be involved in a disorder. Think of them as tiny glowing genetic searchlights targeting one exact spot.
So you introduce these glowing probes to the cells.
Right. And if that target DNA sequence is present and normal on the chromosome, the probe zips right on, hybridizes, and sticks perfectly. And under a special microscope, you see it light up, like a tiny bright beacon showing its exact location.
Wow. But what if there's a problem? Like something's missing or moved?
That's the power of it. If there's a mutation, like maybe a chunk of DNA is deleted or rearranged, the probe might not bind properly, or it might bind somewhere unexpected, or maybe it won't bind at all.
So it's like a genetic GPS that also flags if the road is, I don't know, damaged or completely gone.
That's a great way to put it. Yeah, it tells you if it's there and where it is.
So thinking about diagnosis then, what kind of super precise insights can FIH give you that maybe simple karyotyping would just completely miss?
Oh, it's a massive leap.
Hmm.
FIH lets us see much, much smaller changes. Things like microdeletions or microduplications, tiny rearrangements, stuff that wouldn't change the overall chromosome number or the broad banding pattern you see in a karyotype.
So it can pinpoint issues with incredible accuracy.
Amazing accuracy, yes, down to specific genes or parts of genes sometimes. It's hugely important for diagnosing certain syndromes or even tracking changes in cancer cells.
Absolutely incredible. But I guess sometimes the issue isn't just those tiny details, right? Sometimes the problem is, well, bigger, more fundamental.
Exactly. Sometimes the issue is with the overall number or the large-scale structure of the chromosomes themselves. These are what we broadly call chromosomal aberrations.
Okay, so defects or diseases basically caused because the number or structure of chromosomes in a cell just isn't the typical setup.
Precisely. And they generally fall into two main buckets,
Hmm.
numerical aberrations and structural aberrations. Let's tackle numerical first changes in the chromosome count.
Okay, first up, polyploidy. This means changes in entire sets of chromosomes. What does that mean in human terms?
Well, think of N as a single set of 23 chromosomes. A normal human cell is diploid, or 2 in 2 sets. Polyploidy means having 3 complete sets, 3N, which is called triploidy, or even 4 sets, 4N, tetraploidy. That means 3 or 4 copies of every single chromosome.
Wow. And I think a key point here is that polyploidy, this condition, it doesn't actually exist in humans, does it? Why not? Seems like more information should be okay.
That's a really sharp point.
Yeah.
You see it sometimes in plants, they can handle it, but humans, no.
Hmm.
Having an entire extra set, let alone 2, of every single chromosome creates this overwhelming genetic imbalance.
Like an information overload.
Exactly. It's like trying to build something with 3 or 4 copies of every single blueprint page laid on top of each other. It's just too much conflicting or excessive instruction all at once. Our development is so finely tuned that this massive genetic overload is almost always lethal very, very early in development, like embryo death or non-survival. Our system just can't tolerate it.
That makes a lot of sense. A powerful reminder of how precise things need to be. So if polyploidy is out for humans, what's the more common type of numerical change we do see?
That would be aneuploidy. Now, unlike polyploidy, aneuploidy isn't about entire sets. It's a change in the number of individual chromosomes.
Ah, okay. So not the whole library is duplicated, just maybe one extra book, or one book is missing.
That's the perfect analogy. Exactly. So this is where we see conditions like trisomy. That's 2N plus 1, meaning one extra copy of one specific chromosome. Think Down syndrome.
Right, and the opposite.
The opposite is monosomy. That's 2N minus 1, meaning you're missing one copy of a specific chromosome.
Okay, so what's the underlying cause? How does a cell end up with a wrong number like that?
The main culprit is something called non-disjunction. Think back to meiosis, that special cell division that makes sperm and eggs. Normally, chromosome pairs separate nice and evenly.
Okay.
Non-disjunction means they fail to separate properly, they don't disjoin. So one resulting sex cell, say an egg, might end up with an extra chromosome, and another egg might end up missing that chromosome entirely.
And if that egg is fertilized?
Then the resulting embryo will be aneuploid. It will have either a trisomy or monosomy for that specific chromosome.
And there's a known factor that increases the risk of this happening, isn't there? Age.
Yes, absolutely. The probability of non-disjunction, particularly in egg formation, increases significantly with the age of the mother at conception.
Why is that?
Well, egg cells are basically formed before birth and then arrested in meiosis for decades. Over time, the cellular machinery responsible for that careful chromosome separation might just become less efficient or perhaps accumulate some damage. making errors more likely.
That's really interesting. Okay, let's talk specific examples, starting with autosomal trisomies, extra copies of the non-sex chromosomes. The most widely known is obviously Down syndrome.
Right. Down syndrome, which is trisomy 21, the karyotype notation is 47 plus 21, meaning 47 total chromosomes because of that extra copy of chromosome 21.
And the features?
They can vary, but often include certain physical characteristics like a flatter facial profile, maybe a rounder head shape, distinctive patterns on hands, and there's typically some degree of intellectual disability, again, varying quite a bit.
And then there are other trisomies, often with more severe outcomes, like Edwards syndrome and Patau syndrome.
Yes, sadly. Edwards syndrome is trisomy 18, and Patau syndrome is trisomy 13. These involve extra copies of slightly larger chromosomes than 21, and the genetic imbalance is much more disruptive.
So, more significant health issues.
Much more. They often involve severe developmental challenges, major problems with organs like the heart, kidneys, brain structure. Features can include a smaller head and jaw in trisomy 18, or serious heart defects and facial malformations in trisomy 13. Tragically, survival beyond infancy is very limited for both.
Wow. It really highlights how critical that precise genetic balance is. Now, flipping it around, autosomal monosomy, meaning missing a non-sex chromosome. You mentioned earlier this doesn't really exist in humans. Why? It seems almost counterintuitive that having an extra copy, like in Down syndrome, is more survivable than having one missing.
It does seem odd at first, doesn't it? But the reason likely involves something called gene imprinting and just the sheer necessity of having two copies of most genes.
Gene imprinting, what's that?
So for most genes, you inherit one copy from your mom and one from your dad, and both copies are active. But for a small number of crucial genes, only one of those copies is active, either the maternal one or the paternal one. The other copy is deliberately silenced or imprinted.
Okay, so only one working copy for those specific genes.
Exactly. Now, imagine you have a monosomy. You're missing one chromosome entirely. If that missing chromosome happens to carry the only working copy of one of these essential imprinted genes,
Then you have no working copy at all for that vital instruction.
bingo. The whole developmental process just hits a wall. It can't complete properly without that key instruction, leading to non-survival very early on. Having an extra copy, like in trisomy, causes problems due to dosage imbalance, but it's generally less catastrophic than having a vital blueprint page completely missing.
That clarifies so much. Okay, let's shift gears slightly to the sex chromosomes X and Y. Aberrations there are often, well, different in their outcomes. Let's start with Turner syndrome. Karyotype is 45X.
Correct, 45X. That means only one X chromosome and no second sex chromosome, no Y, no second X. This only affects individuals who would otherwise be female.
And the characteristics.
They often have issues with sexual development, like poor breast development, maybe absent menstruation, leading to infertility. They're also typically shorter than average and can have specific health concerns like kidney or heart problems. Sometimes learning difficulties too, but often intelligence is normal.
Now, for diagnosis, besides the karyotype, you mentioned bar body analysis is important. Can you remind us what a bar body is?
Sure, in a typical female cell, XX, one of the X chromosomes gets largely inactivated early in development. It compacts down into this tiny, dense structure you can see under a microscope. That's the bar body. It's a way of balancing the dose of X chromosome genes between XX females and XY males.
So, a normal female cell, XX, has one bar body.
Right. But in Turner syndrome, 45X, there's only one X chromosome to begin with. There's nothing extra to inactivate. So you find no bar body. It's a helpful diagnostic clue.
Fascinating. Okay, what about males? The most common sex chromosome condition there is Klinefelter syndrome.
Yes, Klinefelter syndrome. The karyotype is 47,000 XXY. So these individuals are male because they have a Y chromosome, but they have an extra X.
And the effects.
Often includes things like reduced male secondary sexual characteristics, maybe less facial or body hair, sometimes increased breast tissue called gynecomastia. The tests are usually underdeveloped, which commonly leads to infertility. They might also be taller than average, and sometimes there can be learning difficulties or increased risk for things like type 2 diabetes later on.
And the bar body test here, how does that work out?
Well, a typical male is XY, no extra X, so nothing to inactivate, no bar body. But in Klinefelter, 47,000 XXY, there is an extra X chromosome. So one of those X's does get inactivated.
Meaning you do find a bar body in a male cell.
Exactly. Finding a bar body in cells from someone phenotypically male is a strong indicator of Klinefelter syndrome.
That's a neat diagnostic trick. Okay, briefly, there are others too, right? Like triple X syndrome.
Right, 47,000 XXX, an extra X in females. Often this has very mild effects, maybe just being taller than average. sometimes slight delays in speech or motor skills, but many individuals might not even know they have it. Diagnosis. You'd expect two bar bodies here because two X's need inactivating.
And Jacob syndrome?
47,000 XYY. An extra Y chromosome in males. Again, often quite rare and frequently has minimal noticeable effects, maybe just taller stature. Sometimes linked, controversially in the past, to behavioral issues, but that's largely debunked. No bar bodies here as there's only one X.
So for all these numerical issues, Down syndrome, Turner, Klinefelter, what about support and treatment? You mentioned diagnosis via karyotyping, often prenatally.
Yes, prenatal diagnosis through amniocentesis or CVS is common for detecting these early. Postnatally, it's usually recognizing symptoms and confirming with a karyotype.
But is there a cure? Can you fix the chromosome number?
Right now, no. There's currently no cure in the sense of going into every cell and correct the chromosome number. That kind of gene therapy on a massive scale is, well, far beyond our current practical abilities.
So the focus is on support.
Absolutely. Medical care to manage any associated health symptoms like heart issues in Turner or Down syndrome. Hormone replacement therapies can be really important, especially for Turner and Klinefelter syndromes. Speech therapy, physical therapy, educational support for learning difficulties. It's about managing the condition and maximizing potential.
And genetic counseling must be vital for families.
Hugely important, helping parents understand the condition, what to expect, the prognosis, and critically, understanding the risks, if any, for future pregnancies.
Okay. So we've covered changes in chromosome number, but you said there's another whole category, structural aberrations.
That's right. Here, the total number of chromosomes is usually normal 46 in humans. But the problem lies in the actual structure of one or more chromosomes. Something's broken or maybe rearranged, or a piece is missing, or there's an extra piece within the chromosome itself.
So the count is okay, but the blueprint pages themselves are torn or copied wrong or stuck in the wrong place.
That's a good way to think about it. They're caused by large-scale mutations, often involving breaks in the chromosome and then maybe faulty repairs. Let's run through the main types. First, deletion.
Losing a piece.
Exactly. Loss of a segment of a chromosome. If the deleted piece is too big, say more than about 2% of the total genetic material on that chromosome, it's often lethal. A classic example of a survivable deletion is Cri-du-Chat syndrome.
French for cry of the cat, right?
It is. Caused by a deletion on the short arm of chromosome 5, we write it as 5p. Infants often have this distinctive high-pitched cry that sounds like a kitten, hence the name, plus other features like a small head, intellectual disability.
Okay, so deletion is losing a piece. Then there's duplication.
The opposite. Here, a segment of a chromosome gets accidentally copied, and that extra copy gets inserted right next to the original segment. So you have an extra dose of just that specific chunk of genetic material on that one chromosome.
Any examples?
One example is a specific type of Charcot-Marie-Tooth syndrome, a nerve disorder. It can be caused by a duplication on the short arm of chromosome 17. Leads to progressive muscle weakness, particularly in the legs and feet.
Okay, deletion, duplication. What about translocation? That sounds like moving pieces around.
It is. A segment of one chromosome breaks off and attaches to a different non-homologous chromosome. It's not like crossing over, which happens between matched pairs. This is a swap between completely different chromosome types.
Are there different kinds?
Two main ones. Reciprocal translocation is like a mutual exchange. A piece breaks off chromosome A and sticks to B. A and D, a piece breaks off B and sticks to A. They just swap segments. Often the person carrying this is fine because they still have all the genetic material, it's just rearranged. But it can cause problems when they make sperm or eggs.
And the second type?
Is Robertsonian translocation. This is a special type that only involves specific chromosomes called acrocentric ones. Those where the centromere is way up near one end, giving them one very long arm, the Q arm, and one really tiny short arm, the P arm.
You mentioned chromosomes 13, 14, 15, 21, and 22 earlier. Those ones.
Those are the ones. In a Robertsonian translocation, two of these acrocentric chromosomes break near their centromeres. They lose their tiny short arms, which usually don't carry essential genes, and then their long arms fuse together end to end.
Creating one big fused chromosome, so you end up with fewer chromosomes total.
Exactly. You'd have 45 chromosomes instead of 46. But since most of the essential genetic material from the long arms is still present, the person carrying this balanced translocation might be perfectly healthy.
But can it cause problems down the line?
Yes. A common Robertsonian translocation involves chromosomes 14 and 21 fusing. Someone with this is healthy, but they can produce eggs or sperm that lead to translocation Down syndrome. The child gets a fused 14-21 chromosome plus a normal copy of 21, so they effectively have 3 copies of the long arm of 21, causing Down syndrome symptoms. The key difference is the underlying cause is structural, not just an extra-free chromosome 21, important for genetic counseling about recurrence risk.
Wow, that's complex but crucial. Okay, another structural change. Inversion, flipping a piece.
That's it. A segment of a chromosome breaks off, flipped 180 degrees, and then reattaches in the same spot but inverted. The genes are all still there, just in a different order within that segment.
Does the location matter?
Yes. A paracentric inversion happens entirely within Y arm, doesn't include the centromere. A pericentric inversion does include the centromere. The breaks are on opposite sides. Pericentric inversions can actually change the relative lengths of the chromosome arms.
Do inversions cause problems?
Sometimes, like translocations, the carrier might be fine. But during meiosis, when the inverted chromosome tries to pair up with its normal partner, it has to form loops to align properly. This can lead to unbalanced gametes, sperm, or eggs with deletions or duplications, potentially causing infertility, miscarriages, or syndromes in offspring. For example, certain inversions on chromosome 9 have been linked to issues like Walker-Warburg syndrome.
Okay, nearly there. A couple more rare types, isochromosomes and ring chromosomes.
Right. An isochromosome is basically a chromosome with two identical arms,
Yeah.
either two short P arms or two long Q arms. It usually happens when a chromosome divides incorrectly across the centromere, crosswise instead of lengthwise, during cell division.
So it's like deleting one arm and duplicating the other.
Exactly. It results in a monosomy for the genes on a lost arm and a trisomy for the genes on the duplicated arm. Very rare, usually leads to significant health problems.
And ring chromosomes. Sounds like what it is.
Pretty much. A chromosome forms a ring structure. Usually happens if there are breaks at both ends of a chromosome, losing the very tips, the telomeres, which are like protective caps. Without those caps, the broken ends get sticky and confused together.
Problems.
Yeah. First, genetic material is usually lost from the tips. Second, the ring structure itself can be unstable during cell division, leading to further problems. Ring chromosome 14 syndrome, for instance, is associated with seizures, intellectual disability, and other developmental issues.
Okay, wow, that covers a huge amount on numerical and structural changes. But hang on, there's one more really important concept, isn't there? Sort of a blend. Mosaicism.
Ah, yes, mosaicism. Crucial concept. This is when an individual has more than one genetically distinct cell line within their body, so not all the cells in that person have the exact same set of chromosomes.
Like a patchwork quilt of cells. Some normal, some maybe with an aberration.
That's a great analogy. A patchwork or a mosaic. You might have some cells that are typical 46XX or 46XY coexisting alongside cells that are, say, 47 plus 21, trisomy 21, or maybe 45X Turner.
How does that even happen? Is it inherited?
Generally, no, it's not inherited like most of the syndromes we discuss. Mosaicism usually arises after fertilization, during the early development of the embryo or fetus. It happens because of an error in cell division, like non-disjunction, occurring in just one or a few of those early dividing cells.
Ah, so that one cell makes a mistake.
And then all the cells that descend from that one founder cell will carry that same mistake, while other cell lineages in the body that started before the error remain normal.
Okay, so you end up with this mix. Considering how mosaicism develops then, how might the symptoms or, the severity of a condition differ in someone who's mosaic compared to someone where all their cells have the condition? What do you think?
It sounds like it could lead to a real spectrum of effects, doesn't it? The symptoms might be much milder, or maybe they only affect certain parts of the body.
Depending on which tissues ended up with the affected cells and how many affected cells there are.
Exactly. It often leads to a milder presentation of the condition. The severity really depends on which tissues have the abnormal cells, e.g. brain versus skin, and the proportion of abnormal cells versus normal cells throughout the body.
So, for example, mosaic Down syndrome
Often presents with milder physical features and potentially less severe intellectual disability compared to full trisomy 21, where every cell has the extra chromosome. Mosaicism really adds this layer of complexity and variability to genetic conditions.
Okay. So as we wrap up this deep dive, wow, we've covered a lot. We journeyed from understanding that basic snapshot, the karyotype,
Through the power of banding and fish to share to zoom in on the details.
and then really explored those broad categories of numerical and structural aberrations, plus this Fascinating concept of mosaicism.
And it's crystal clear these aren't just, abstract biology terms. They have profound real-world impacts on human health, on development, really shaping lives in significant ways.
Absolutely. The ability we have now to actually visualize and analyze these fundamental blueprints. It's a cornerstone of modern genetic medicine, isn't it? Helps us grasp the incredible precision life requires, but also maybe the resilience of the human body sometimes.
Definitely. And thinking ahead, as we keep mapping the genome in more detail, developing even smarter tools, maybe advanced sequencing, AI helping to analyze these complex images, what new frontiers could open up? Yeah. And understanding, maybe even eventually addressing these conditions.
What do you think? Earlier detection.
Perhaps much earlier, more detailed detection, yeah?
Yeah.
Maybe a better understanding of why specific aberrations happen in the first place, which could potentially lead to, I don't know, new preventative insights down the line. Though, we have to be realistic, actual cures for established chromosomal conditions, correcting every cell, that remains incredibly challenging.
And of course, with that growing knowledge comes, well, ethical questions, right? As our ability to detect these things earlier and earlier improves, what are the complex ethical considerations we need to grapple with as a society? Questions around screening, choice, potential interventions. It's not simple.
Not simple at all. A lot to consider there.
Definitely a lot to think about as we look towards the future of genetic understanding. Thank you for diving deep with us today on this really intricate but incredibly important topic.